We're going to go ahead and get started. It's a real pleasure to introduce Jan Spacek. Did I pronounce that correctly?
Perfect.
Okay, good. Jan, I need to read this. Your credentials are so kind of intricate that I need to read it to get it straight. Jan got a PhD at the University of Spaček in the Czech Republic. And he's currently at the, it's called, the abbreviation is FAME, but he's a senior scientist at the Foundation for Applied Molecular Evolution, which is actually over in Alachua. This is an institute that probably hardly anybody in this room is aware of, but it is a very active astrobiology institute that is has several very renowned scientists, including Jan, working there. So it's a place to be aware of, and it's an important important institute for astrobiology. So Jan got his PhD and then actually has a very interesting and complicated life history. He's basically done research in Turkey, University of California, San Diego, Oregon, the Czech Republic, of course. And so he's really kind of, has a fairly broad scientific experience in astrobiology. But in addition to that, he's also done some interesting things, and you can read the more detailed biography that I sent around. But he bicycled across Australia and, around the Baltic. So this guy is, multifaceted, and he's also working on instrumentation that will hopefully precede Americans, or not Americans, but humans landing on Mars in order to look for life before we actually contaminate Mars. But today he's going to talk about Venus and the mystery associated with these dark, mysterious cloud features that we see. And take it away.
Thank you, Charlie, for inviting me and for an introduction. You actually managed to do it without reading your cheat sheet, so that's great. Okay, I made a little bit clickbaity title to get you all in, and it looks like I succeeded. So I will be talking about Venus clouds, and just a reminder. Oh, it's not moving. Let me restart the share then. Oh, I need to be here. Okay. Give me a second. Restarting the share on Zoom. Okay, so it's complicated, but I got it. Yep. Okay, so this is Venus, as you know, and I will be talking what's happening in the clouds in the middle layer over here between, let's say, 50 kilometers to 70 kilometers. You have this thick layer above Venus of clouds. They are made of concentrated sulfuric acid. And if you look on the Venus, it looks like this. But this is not how you would see it with your own eyes. This is actually at 265 and 283 nanometers. So it's UV. So this is processed image. This is not how you would see it when you look at it. So US astronomers know how these images work. We observe Venus in a UV, infrared, and you get different features at different wavelengths. But I'm, for now, interested how it looks like if you look at it with your own eyes. This is, again, ultraviolet image. And these features, they are rotating on Venus. And it has been shown that they are upwelling from a deeper cloud. And then, so the deeper cloud is darker, it gets upwelled. And on top, the cloud is renewed from photochemical processes. So pure sulfuric acid is not light absorbing. When you bring it from below the cloud, you make these features that are darker, and we can see them on the surface of the cloud. Another image, another composite, and this time they try to make it look like more how you would see it if you actually fly by and look at it with your own eye. And this might be like how it would look like. But all of these images, they were recreations of UV spectra, so you have to trust some astronomers. I'm sure that you all observed Venus much more than I did. So if you look on Venus with your own eyes or with telescope without adjusting for UV, you would see that it's pale lemon yellow. I contributed to observation as well. I confirmed that it's a little bit yellowish. So that's my observation, my contribution to this. I'm not astronomer. I'm a biologist, and I'm standing on shoulders of many astronomers and people who worked before me, of course. First observations of these markings, which I just showed you on the top, were done nearly 100 years ago. Frank Ross in 1928 or 1927 showed that there are these markings, and he explained it that you have like a white cirrus clouds above yellow atmosphere. That model was later disapproved. The gases in Venus, they are contributing to the absorption, but they cannot explain those dark markings. So Carl Sagan, of course, was, I guess, the most famous astronomers observing Venus, and he suggested it might be astrobiology target, but at the same time, he said that there are no organics in the clouds of Venus. And then other researchers in here in this lineup located the absorber within the cloud layer and showed that, most of the, or half of the sunlight that hits Venus is actually absorbed because of that unknown absorber, which is quite interesting. And yeah, the last Jan Yoli in this lineup, that's a collaborator. I work on the results I'm going to present to you. So alternative version of this talk is a beer model of Venus clouds. So I thought that I will show you this experiment. Here I wanted to make a nice cloud using this, this is like nebulizer, which is using ultrasound. And I would ask some volunteers to come over and check it, and because it looks completely white mist. And I would have you guessed which color, let me show it to the Zoom people, which color of this lineup is actually, making the fog in here, and you would not be able to guess it because it's completely white. And then I would shock you all by showing that it's completely black. So even if you have completely black liquid and run it through a nebulizer, make submicro or micrometer particles, the fog look completely white. So unfortunately, I think I killed it because this is supposed to be run with clear water, and I'm running it with saturated solution of acrylic paint, which is color, used for painting. So I think I killed the ultrasound transducer on top. But to make you more familiarized with it, you've all seen people vaping. It would be great for marketing if you can make pink fog, right? Nobody has done it because it's impossible. If you have very small particles, no matter what they are made from, the absorption, the contribution of the scatter or the color by the light, by the absorption within the bulk liquid is minimal. So even if you run this through a nebulizer, it's completely white. So why it's beer model? Let me demonstrate. This is campus-friendly non-alcoholic beer. So empirical data. Oops.
We would have gotten even a bigger crowd of students.
Right. Yeah. This is beautiful astronomy talk. Okay, so here's the experiment. Right, we have the foam, which is made from the same material as this, but because we have a lot of interfaces between gas and liquid, then the scattering is influencing how it looks like. So even if you have a dark beer, dark beer can have also very light foam, as again, because of the scattering. And so if you want to find if you are going to drink a light beer or dark beer, you know, just looking on the foam doesn't give you all the information. So yeah, that was for this part, right? If you have a red color, it's blocking the absorption is in the blue part of the spectrum. You have the blue pigment over there. It's blocking all the red light. And then the total height of the absorption indicates, or the absorbance per centimeter indicates concentration. Unfortunately, this experiment failed. So important point, if you have scattering on air and liquid interface, that contributes more to overall appearance than the material which is made from, if the particles are small enough. And yeah, you cannot determine from remote observations what's the liquid color. Another nice example, you can see a river which is dark about coffee with milk. If you run it through a waterfall, make a mist, that's again white. Again, example of this. So for a century, people were thinking, why is Venus yellow? But I think the better question is how dark the liquid is to make the clouds appear a little bit yellow. And from what I showed you, so we know that particles are very small. So just from using this logic, you would assume that the liquid must be very dark to contribute at least something to the overall spectrum we observe, because most of the light is just reflected from the surface, but just small fraction is penetrates into the particle and is absorbed. So, and from this, I cannot tell if because I haven't done any modeling, I cannot tell whether it's the liquid is dark as this beer, or it's a black Guinness, or if it's dark as tar. And I was first to ask this question, actually, so that was very surprising to me. And this lady from South Korea, Yeon Yo Lee, helped me made a model. She specializes in observation of Venus and radiative transfer modeling. So she provided her time and give me answer. And here's the answer. Yeah, that's not good. Okay, the answer is super interesting, because the important part here is the absorbance per centimeter, right? It's very high. And from August Beer, again, when it's beer talk, he tells us that the absorbance at a given wavelength is dependent is a logarithm of light, incident light divided by the transmitted light. So you are shining, basically, if you shine light through this is about 1 centimeter. So the light which passes through, that's what you observe. You make a ratio of these two numbers, and this gives you absorbance. And so if you check the number of or amount of transmitted light, so here is the absorbance because it's logarithm. So the percent of transmitted light, it drops very fast. So with your human eye, you can tell difference between absorbance 3 and absorbance 3000. To you, it looks completely black or completely dark for a given wavelength. So if you see absorbance 3000, that's completely black. If you have just 1 micrometer of it, this is basically very thick paint. I sacrificed my blood for this experiment. I prick my finger, diluted the blood 1,000 times, and measure its absorbance, then multiply it again by a factor of 1,000, and compare it with the absorbance of the Venus clouds of the model light, or model absorbance of the bulk liquid. And you can see that Venus absorber at its maximum is absorbent more than my blood at its maximum, which is super interesting. I'll explain why. Another thing which August Beer is telling us that from absorbance, we can calculate the concentration of material in a solution. So the difference between these two is that here we have about 100 times less of the material, and you can see that this is darker than this because this simply absorbs more of the red part of the spectrum. And so if we know the extinction coefficient, epsilon, we can calculate the concentration. And so, oh, another thing. When I was looking for August Beer, I found this. This is like foreshadowing that, no beer, no life. That's an astrobiology signature. If you don't have a beer model, you will not find life in Venus. Anyway, if we look on the extinction coefficients, we have some I picked very well absorbing molecules. What you notice in these pigments that they all have conjugated double bonds, which work as like little antennas that can absorb light. So basically, photon can excite these p orbitals, molecular orbitals to a high level, get absorbed, and that's why we see the absorbance. The important part in this chart is that if you take, for example, the chlorophyll with a known extinction coefficient, you can calculate how much of the chlorophyll you will need to have in a per liter as a concentration to satisfy absorbance around 3000. So you need about 20 grams per liter concentration of chlorophyll to replicate the absorbance we see in Venus clouds. And that's comparable to what you see in plants. So the material above, on average, Venus clouds, if you collected the liquid would be as dark as, 3 times darker than my blood, or about as dark as plant material, which is very surprising. Another thing, ferric chloride, this FeCl3, was suggested as the best candidate for the absorber so far in the clouds. But if you check its extinction coefficient, it's much smaller than these other absorbers. So you would need 3000 grams per liter, given that ferric chloride has a, how we call it, density about 2000, you will need more than 100% of ferric chloride just to satisfy the need for the absorbance. So if our model is correct, it excludes ferric chloride as being the pigment because it's simply not absorbing light enough. Another thing, the astronomers who propose ferric chloride likely never put ferric chloride in sulfuric acid, because if you do that, you find that it immediately turns to ferric sulfate. And the sulfate is completely, it's a precipitate which is white or whitish, so it absorbs even less light. So I don't think it's ferric chloride. So the red line in here is again the modeled Venus absorber, or absorption we expect from in Venus clouds in a bulk liquid, modeled by Jan Yoli, and it's compared to bacterial chlorophyll. Again, we are not saying that there is bacteria making bacterial chlorophyll, but you can find molecules that have very similar absorption profiles. And this is just to inspire your imagination how these molecules would look like. It would be a large molecule. Again, the wavelength tells us something. How large is the overall molecular orbital, which is doing the absorption, and then intensity. Well, the absolute value of the absorbance is tell us about how concentrated it must be. So
Beyond in that slide right there. In chlorophyll, you've got a lot of absorption way up above 500 nanometers.
Yeah.
And so that would be also something that one should be able to observe, right?
So I discussed that in the paper which you have over there. By the way, I have 2 more copies if you and other people want a paper which will soon be in astrobiology. I was hoping it will be already published. So 2 more copies if you want to read them. Yes. So normally with porphyrins, you have 2 absorption maxima. One is in that UV blue part of the spectrum, and another one is in the red part of the spectrum. But some of the porphyrins have the second band. I think it's called Soret band. You know it better, right?
Soret.
So that Soret band can be much lower or distributed over a longer band. So if it is porphyrin, I'm not saying it is, there are porphyrins which have very diminished Soret bands depending on the side chains, what's hanging here and here and how symmetrical are. Yeah, it's whole science which you can study somewhere else. So again, the team members that helped me write this paper basically satisfied my curiosity how dark the Venus clouds are, or the bulk liquid is. There's a Jan Lee, Janos Petkovsky, Paul Rimmer, and yeah, so now we have this conflict, right? We have astronomers since 60s. They were saying that there can be no free carbon, hydrocarbons, formaldehyde, or any other organic molecule present in more than trace amounts above Venus. So that's those guys. And then we are saying that our model shows that we need about 10% of mass of the cloud aerosols to be the pigments, which are very highly absorbing and which very likely are not inorganic absorbers, because no inorganic absorbers we expect to be in the clouds are absorbing enough to satisfy that model. So we have a battle to come when this is published. I expect a lot of pushback against that because people assume that it's very chloride and it's sort of solved, but it's not. So is there a justification for organics in the clouds at the amounts what we are proposing? So are there organics in clouds of Venus? Definitely yes, because they are falling in from space. You have meteorites and micrometeorites constantly falling in the Venus clouds. We put a little bit of Murchison meteorite in a sulfuric acid and heated it up a little bit in an oil bath. And basically the organisms got leached from the meteorite and then reacted in the sulfuric acid to produce this yellowish coloring compared to the control, which stayed clear. And under black light, you can see that the organics are also fluorescent. So yes, but this info that would not be high enough concentration. We need tons of it. I mean, there are tons of it in Venus, but we need a lot more. So the question is there another way how to produce locally in the clouds organic materials? And this is something I started working on in 2021. And in 2023, December 2024 was actually published. Was this paper, how you produce organics in clouds of Venus. So these are people who work on this. Gage, Spencer, me, Stephen Bennett, that's the boss in Alachua, working on synthetic biology, life origin, now also biology. Some of you know Daisy Sharma. She worked with us, and this is Paul Rimmer from England. And so this is the model which I proposed in 2021. You have water coming from volcanoes, as Charlie pointed out before, and you have organic carbon generated from photochemistry. So you can generate small amount of formaldehyde from this photochemical production from carbon monoxide and hydrogen, or actually H2S. Paul Rimmer have made this part of the model. So you are generating some organic carbon, which itself is not light absorbing, but as it reacts further, it turns into larger and larger molecules. They cross-link, make this tar or black molecules. And if you remember from the first slides, if you have some upwelling from here, bring it up, then you see those dark streaks in the clouds. So that was what I proposed in 2021. And then in 2024, we've done the modeling to see how much you actually produce of this material. I'll get deeper in the chemistry in next slides, but for now, if you put formaldehyde in concentrated sulfuric acid, there was a big surprise. You turn it into this dark liquid, and then if you dry it, you get to this black carbon. So the assumption is that as things are falling through the clouds, if they get to the bottom of the clouds where things evaporate, it will evaporate the sulfuric acid, and you will be left with that black carbon falling through the clouds, which then at the bottom of the close to the ground where you have 90 bars and close to 500 degrees Celsius, you will turn it to carbon monoxide through Boudewald reaction from using carbon dioxide as oxidizer actually. So it's important to know that sulfuric acid has very high affinity to organics. You might know that sulfuric acid is hygroscopic, which is taking in water, but it's also taking in formaldehyde and other small volatile organics, which concentrate. And then when they are present in those droplets, they react further. And so contrary to general health assumption, those organics are actually, well, they are not stable than what we are producing, but they are not destroyed. They are turned into larger organic molecules until they finally turn to that black carbon that I showed you. And actually our friends in MIT, Sarah Seeger and her team, including Jan Spacek, showed that many of these organics or molecules that you have in your bodies, like nucleobases, some amino acids, and other small biomolecules actually survive in concentrated sulfuric acid. So that's super interesting. But back to the paper. So the yellow liquid, which I showed you, I called it red oil because that's how they called it in petrochemical industry. When you are making alkylation in sulfuric acid, when you are making gasoline, you want to make octane. So you take your shorter molecules like isobutane and butane and run it through alkylation reaction in concentrated sulfuric acid to make octane, which then you use as gasoline. So that was my literature, which I used to make this model. So again, and this literature was known. The reason that people did not discuss this was that, I guess, they didn't read petrochemical papers. So, and yeah, and so this would turn into that black tar, and yeah, and it's nicely fluorescent. So again, this formaldehyde, if you put it under UV light, it's usually green or blue, but sometimes you get this nice yellow or golden fluorescence. And what we've done a lot of was mapping out how the fluorescence look of different organic materials when you put it in sulfuric acid. And so you have these nice lights coming from it. So again, these are concentrated sulfuric acid solutions with different organics, which were originally not fluorescent, but once you put it in sulfuric acid, it reacts, cross-link, make larger organic molecules, which are fluorescent themselves. And this is the second part where you heat it enough, the molecules keep cross-linking, making larger molecules until you turn to that black carbon. And so in the 2024 paper, we mapped actually using isotopically labeled carbon-13. We mapped how you get from formaldehyde as the single carbon molecules to glycolic acid, which is 2 carbon molecules, which is a very important step. And then when you have two molecules, two atoms, two carbons, you get to larger molecules. So I will not go into details in here or here. We can discuss it later, or you can read the paper. So that was one way. You use formaldehyde to produce reduced organic carbon. There's alternative way, which I believe exists.
So, but you mentioned that the you get some of the products are the fluoresce.
Mhm.
Is that something you might expect to see in Venus? If
Yes, I'll get that.
okay,
Yep, exactly. That's what we plan to do.
I haven't read your paper yet. I'm just
Okay. So what you can do, you can take sulfuric acid and you can take formic acid, mix them together, and then commit suicide. Suicide by inhalation of carbon monoxide produced by mixing formic acid and sulfuric acid with confined space is a rare method of suicide, so don't do it. That's how the reaction normally leads to carbon monoxide is dehydration. You will just remove water from the formic acid. Again, because sulfuric acid is so hygroscopic, it just rips the carbon monoxide from it. So I asked Victor to do these reactions for me to, save my life. What's super interesting is that sometimes you get some product which is not just carbon monoxide. So this reaction, production of carbon monoxide, happens in a room temperature, but if you cool it down, the carbon monoxide, even if it's generated in sulfuric acid, it does not bubble out. So I just put it in a freezer, let it there for a week. And then when I opened the freezer, it was brown and fluorescent. So something happened. And here's a movie. You can see that. Oh, so then I took the tube and let it warm up. And you can see over here, you have a region where some gas is produced. And above it, you have a region where it turns brown. And then after it actually burned through this, like going down, it was super interesting. So you have like a layer where it's burning down. So it suggested some autocatalytic reaction where the products catalyzing the further production of the brown stuff, which is fluorescent. And I don't know actually what's happening there. I assume that's another disproportionation. Instead of dehydration of the formic acid, we have some sort of disproportionation, similar to what you have in Boudoir reaction, where you take carbon monoxide, carbon monoxide and disproportionate it to carbon dioxide and carbon. So something similar happening there. I didn't manage to replicate it. When the reaction burned to the end, it was all nicely fluorescent. So if you want to describe what's going on in there, you would be very famous. You might change how we see Venus, because in clouds of Venus, we have sulfuric acid and ton of carbon monoxide. And carbon monoxide is basically hydrated, formic acid is hydrated carbon monoxide. So maybe we are getting those products in there. So did we solve the mystery of Venus clouds? Nope. So not yet. The amount of organic material which we modeled in this paper, we show that we are producing like millimolar concentrations or sub-millimolar concentrations of reduced carbon. So we are like 4 orders magnitude below the amount of the organic material which we predict in this year paper. And the absorption profile does not match. As you remember, this brown stuff, it's absorbing throughout the entire spectrum and has a very similar spectrum to graphene oxide. And I think that's what we are producing with this. So you get to very complex molecules which are absorbing in the entire spectrum. What we see here is some molecule which is more organized. If you remember the bacterial chlorophyll, It's not a random mixture of large molecule. It's a specific pigment, actually, what we see. So, but if you take this formose reaction, that's how you get from formaldehyde to larger sugars, and you give it enough time and evolution, you get to life as we know it, right? So maybe what we are predicting here is not what's happening right now in the Venus. Maybe what we are predicting is, the primordial chemistry which was happening before. It can still be happening, but if you have enough time, there could have been some selection towards, because you can have, for example, particles that contain absorber with this spectrum are more likely to persist in the atmosphere and then, conduct some selection. Maybe if you have evolution, you can get to Venusians, which would be sub-micrometer to stay in the clouds. Also notice that they have a blood made of sulfuric acid, so that's very relevant. And yeah, again, I would make a reference to my beautiful experiment here, that if you want to really find out, you cannot do remote observations, you need to get there. So Sarah Seeger and her team, which I joined, proposed to send a little probe, this small probe, which shoots a blue laser and observes green fluorescent light. It also observes, so that's fluorescence, but it would also observe reflected light to analyze the properties of the droplets. So if we detect fluorescence, it means that we find organic material, and then with this experiment, it would be more yes/no question, is it there or not? And I suspect very much that we will see it. And then with later experiments, we maybe get deeper, like spectrum of the fluorescent light. Anyway, yeah, and the experiments we've done with Victor and other students, they informed which laser will be selected. So we are aiming for excitation at 440 nanometers and detection for, I think, 480 and more. We remove this other filter, so we are detecting all this fluorescence. So the data we gathered informed how the mission is done. And there is more. I told him, so Janusz Petkowski is another team member from Sarah Seeger team. So he shared with me some exciting news. And I was like, yeah, I'm going to tell it on a seminar. That was going to be so excited. And Janusz said, no, let's not share it yet. So stay by for no, but it's ongoing. We have new results, new exciting things. And so you might know Wayback Machine, which it's like Internet Archive. What you might know, there's a way forward machine, which goes forward in the internet history. So you can search for the coolest science in the future, and then you load the Internet of the future, and it will show you that University of Florida astronomy team discovered life in Venusian clouds. It's a team led by Charles Telesco discovered that the Venus unknown absorber is a chiral molecule likely produced by alien biology. Yeah, but we already beat you with finding life on Mars, so.
I didn't put you up there.
No, and not even for this slide. So
Yeah.
I'm not saying it's aliens, but if it's aliens, they will very likely, the absorber will be likely a chiral molecule, which would be causing the circular polarization. So yeah, there you go. This is my last slide. So I'm happy to answer any questions until evening. Thank you.
Any questions?
All the chemistry you're talking about happening is happening in the liquid phase, right? It's all in droplets, like
So-
no gas phase. Is that right?
Yeah, and we just on this paper. Yes, it's It's both. So you have photochemistry happening in the gas phase. We didn't test that in a lab. We modeled it with Paul Rimmer. So he done the photochemical modeling where you produce molecule up to formaldehyde. The reaction afterwards, getting from formaldehyde to glycolic acid and large molecule, that's in a concentrated sulfuric acid. Yes.
So part of my reason we're asking the question is I'm curious what constraints you get from millimeter spectroscopy through the atmosphere, like the phosphine detection claim. That's one possible line of detection. There are other, I think there are other lines of detection. Can you do they give you any useful constraints on what might be happening in the gas phase?
So
in parallel with liquid phase?
yeah, I don't know. So from gas phase studies, they were making those conclusions that you have if there's organic material, it's going to be trace amounts. And I suspect this because the sulfuric acid is has very high affinity towards all volatile organics. So you have two factors. First, you concentrate the organics in the particles. Second, if you are making large organics, they are not going to be volatile, so they don't get out. And then the spectra you observe in the gas phase, I don't know how well you map the molecules in the liquid phase if they are not in the gas phase, if you understand what I'm trying to say. So if I expect there are absorbers in the liquid, the gas phase observational astronomy cannot maybe miss that. But I'm not an astronomer. Maybe you find some way how to figure it out.
Yeah, Chris.
So if the autofluorescing nephelometer is able to detect the organics that you're hoping with the Morningstar mission, what would be a proper follow-up? And if you end up saying that it is alien, then you're saying it's not alien, what would be the life detection instrument or experiment that you would like to see in the classifications?
Well, yeah, it's actually the Morningstar missions, missions led by Sarah Seeger. They have already a lineup of subsequent missions. So if we see some fluorescence in the clouds of Venus, then we will likely get funding for subsequent mission, which is a balloon, which has I think spectrometer and does other things. So analyzing the droplets in situ. And the third mission would be to collect it and bring it back to study here. And yeah, as I'm saying here, detection of circular polarization, which you can do from Earth, would be a way how to test this. And in situ, of course, maybe some microscopy, because you can actually collect it.
Yeah, and Chris is working with us in the lab to build an instrument that will hopefully do that, maybe from one of the UF observatories. I had a question. So would we be able to detect the fluorescence from Earth? In other words, or is the atmosphere between the layer fluorescing and the upper layer, is it, the outside, is it basically too thick? Is it?
No, I had the same questions. Yeah, I know. I agree that this is a good question because I also keep asking that. So how you would observe it is because you would not use laser to excite it from Earth. You would use sunlight to
UV excited.
Right. So you would have excess of light coming as a reflected light. So let me get back to
reflected out somewhere in the visible, right?
Yeah,
that's where your fluorescence.
Yeah. So that's the interesting part. So come on, where is it? Yeah, let's do. Ignore the purple line and imagine that this goes forever. So maybe this goes down, not because there is no more absorption. It might be going down because the fluorescence is actually what we are observing in albedo for every given wavelength is the total amount of light we are getting back. And we cannot distinguish whether this light is coming as a reflected light or as a fluorescence. So you will need to make a model which assumes how much light you expect to get back if there was no fluorescence and compare it with the reflected light which we are actually seeing. So maybe there are other bands where you are absorbing, but they might be overlapped with the fluorescence, so you would not see them.
And you think that the reason you won't see, fluorescence usually has a very distinct lines, there's set of lines associated with it, but here you think
No,
it's going to be smeared out. Oh,
that's in a gas phase. If you have a liquid phase, the fluorescence is very broad.
Okay, interesting.
So yeah, even the absorbance, like these spectra are so broad because you have the vibration and effect of the solvent are broadening the absorption lines. If you are observing in vacuum, as your astronomers are used to, you have nice crisp line.
Okay.
Because you have exact quanta where you can absorb, but if you have molecules around, it broadens.
Okay, really interesting.
In both ways.
More questions? Yeah.
So, I've seen, pictures of like rocks and stuff like that for the fluorescent, molecules like, molecular hydrogen can also fluoresce, at a certain energy, stuff like that. What about organic molecules specifically, is making this fluorescent light that you're interested in? Like, what differentiates them from rocks or just other molecules that fluoresce?
Right. So the thing is, would you expect those molecules to exist stably in the clouds of Venus? So yes, you can find minerals that are fluorescent. Usually they are fluorescent because you have some flaws in the lattice, and that's where you see fluorescent. If you actually dissolve that mineral in some solvent, maybe in sulfuric acid, you lose that fluorescence. Hydrogen as a atomic hydrogen, that's what you said?
Molecular hydrogen.
Molecular hydrogen. For what do you need to excite molecular hydrogen and at which wavelength it fluoresces? So at near UV or visible light, I doubt that it's fluorescent. It's in that part, right? It's gonna be super short wavelength to fluoresce with hydrogen, right? I don't know. Tell me, because I never heard about that. That's maybe more relevant to astronomy than to.
I think you need high energies that might not be in.
Yeah, so I think you need to get very short wavelengths, and once you are in the atmosphere of Venus, you are already causing that ionization with those very short UV lights. Basically, you at this deep in the atmosphere, you don't get high enough energy to do that fluorescent, and the emission of the molecular hydrogen would be also very short wavelength, I assume.
Okay, so following this thread, the black stuff you're making, it's PAHs, right? Is that a fair summary? Like they're all PAHs extra stuff.
Yeah.
So PAHs in astronomy, we think of them as things that glow in the infrared. We see them as these excess features there. Is that still happening in these samples, or that's suppressed in the phase? That it's all only thing of them. So the question is, could you look at the infrared to
Look for the pH features, yeah.
Can you? I don't know, because I can't.
Well,
So
JWST could, of course, means it might too bright.
no. Here's the thing. Again, we have it in droplets.
Yeah.
So
that's
again, most of the light you are observing is due to scattering on the surface. Small fraction of it is actually getting inside. That was the whole presentation of these things. And beer. So I don't know. So PA, you can find these molecules if they are suspended as a dust. Can you find it if it's in a droplets? I don't know.
Interesting question.
all right. So get in the spectrometer lab and shine at that stuff. Or
Yeah, I'll give you that stuff and you can do it.
Yeah, sure.
I don't know if it's changed since the last time I read about it, but in my understanding, the nephelometer that will be on the Rapid Lab mission will essentially be in free fall when it's entering the atmosphere of Venus. Doesn't have a parachute, doesn't have anything. My question is, will you get enough signal as it approaches, like, because you're only going to be interested in a 50, 60 kilometer altitude height. Will you get enough signal to actually see the fluorescence that you want to see?
Yeah, detecting individuals as it falls through the clouds, and we've made some models that we'll get I forgot the exact number, many of the detection, many of the reflected light for sure, and the same amount of fluorescent light. So it's split by wavelength. If it's longer wavelength than the excitation laser, it passes to detection of fluorescence. So we should have thousands and thousands of detection events. Then they like process it on board and send the data back to the spacecraft, which does not enter the atmosphere, and then it's transmitted back to Earth. So that's the plan in these papers. And yeah, it will be just couple of minutes in the clouds. So
You don't try to slow it down or anything? I mean, you don't put like little cups out or something that
nope,
act like little.
those few minutes are enough. And this team made the models. Actually, they've flown this nephelometer on a drone through clouds of well, near volcano. So volcanoes produces SO2, SO3, which in the atmosphere turns to sulfuric acid droplets. That's how you get acid rain. But near the volcano, you have a very high concentration sulfuric acid. And because on Earth you have plenty of organics in the atmosphere, the organics get uptaken into these sulfuric acid droplets. So I think they already observe fluorescence from these molecules. So again, on Earth, you are producing those polycyclic aromatic hydrocarbons usually from terpenes, which plants are just putting in the air. But also you have a lot of other organic volatiles in the air on Earth. So yeah, very short time is enough.
Yeah, more questions? We have just a little bit of time for a couple more questions if you have them. This is great. I mean, what amazes me is for years, we just said, we always, when we were talking about astrobiology, we just skipped over Venus because everybody said, Venus isn't going to have any life. It's so, so hot. And now and the sulfuric acid, and all of a sudden there's this enormous competition going on as to who's going to basically get to Venus and either prove or disprove that there's any life there. It's like all of a sudden, and people are putting together small experiments just on tabletops basically to do this, whereas other people have been planning these huge missions to Mars and the outer solar system. So this is really exciting. It's like old-fashioned science. You guys got to get involved in it. And so yeah, tabletop science again. It's fun.
Yeah, when I was starting working on this, I was very skeptical. So I was like, yeah, okay, I'm going to do these measurements for you, but it's going to be all negative results for. So the team, actually, this was kickstarted by the phosphine detection. I still don't know whether there's a phosphine in any quantity in Venus clouds, if it was false positive or not. But what came after, detection of organics. So I started very skeptical that there could be anything fluorescent in the clouds of Venus. But then we've done these experiments I showed you, and I cannot be skeptical anymore. So you have definitely organic carbon in the clouds of Venus. The question is how much, and if the model which we just showed you is correct, and if you have really large concentrations of organics. So you can review those papers, find the mistakes, and maybe this is wrong. Maybe we are right.
Yeah, great. If there are any more questions, Jan will be around all afternoon. He's going to be up in the lab for a while with us. You can, if you're in that area, you can just drop in and join the conversation. Otherwise, he'll be around. And if you want to talk with him one-on-one, feel free to let me know or Jan know. Somebody upstairs.
Here and still kind of not, Doctor. But
The people online, sorry. Didn't even think of it.
Jason.
I'm sorry. Oh, then they're cooking upstairs, yeah.
These reactions you are describing seems to require to be contained with a concentrated environment like the clouds. To proceed to significant levels, would you expect similar reactions to be able to happen in hydrogen, helium dominated warm Jupiter? No, so the reactions we are describing in the liquid phase are actually done or catalyzed by the sulfuric acid. Without concentrated sulfuric acid, you would not see those reactions which I described. So thank you for the questions, but unless you expect concentrated sulfuric acid, you will have different reactions. And you can produce large organic molecules through photochemistry as well. The question is if those are sustained in the atmosphere. We definitely know that you produce a lot of interesting organic molecules on Titan, for example, from photochemistry, even though it's very far from the sun. So,
Sorry, I forgot all about the people online. I apologize, folks. Okay, well, with that, let's thank our speaker again and yeah.